We are thrilled to have you all here. Good morning, good afternoon, wherever your time zone may be. And welcome to today's national webinar and congressional briefing of the Coalition to Advance Maternal Therapeutics. The title of today's webinar is Pregnant and Lactating Populations in Research, How Leaving These Populations Out Leaves Them Behind. My name is Catherine Schubert. I'm the president and CEO of the Society for Women's Health Research, which is a more than 30-year-old national nonprofit dedicated to promoting sex differences in disease and improving women's health through science, policy, and education. Additionally, and notable for today's event, we are the administrative home of the Coalition to Advance Maternal Therapeutics. Before we move into today's program, I want to review just a few details and housekeeping notes for you all. We'd like to thank, first and foremost, the Coalition's corporate members whose support has made this event possible: Biogen, Covis Pharma, Janssen, and UCB. Please note that today's web event is being recorded and will be posted online. We will send out the recording and any additional resources mentioned in the follow-up email to all registrants. And to ask a question to the panelists, please use the Zoom Q&A function. Alternatively, you can use the chat function. We will ask questions at the end of the event following all panelists' presentations. So today's event, as I mentioned, is being hosted by SWHR in our capacity as administrative home for the Coalition to Advance Maternal Therapeutics. And the coalition was founded in 2014 by a group of organizations who recognized the tremendous need to understand the effect of drugs on a woman and her pregnancy, and the ways in which pregnancy might alter the uptake, metabolism, and effect of medication. The goal of the coalition is to better understand the safety and efficacy of prescription drugs, therapeutics, and vaccines used during pregnancy and breastfeeding. It is due to that mission that we're here today and joined by our expert panelists to discuss concerns and considerations for including these populations in research. So with that said, I will introduce our panelists. First up, Dr. Christina Chambers, who's a professor in the Department of Pediatrics, School of Medicine at the University of California, San Diego, Chief of the Division of Environmental Science and Health, and co-director of the Center for Better Beginnings. She is the principal investigator of mother-to-baby pregnancy studies and the UC San Diego Human Milk Research Biorepository, 2 nationwide longitudinal cohort studies focusing on the safety of medications, vaccines, and substances, infectious agents, and other environmental exposures in pregnancy and lactation. Dr. Chambers has been an engaged leader in the topic of inclusion of pregnant and lactating populations in research. Dr. Ann Lyerly is a professor of social medicine at the University of North Carolina, Chapel Hill. She is also a research professor in obstetrics and gynecology. Her research addresses socially and morally complex issues in women's health and reproductive medicine with a focus on how people assign meaning to reproductive events. A central goal of her work is to inform and reframe debates based on the views of those most profoundly affected by them and to appropriately weight these individuals' interests in shaping reproductive health care. And finally, Kara Pollan is the Associate Director for Communications in CDC's Division of Birth Defects and Infant Disorders. In this role, she leads efforts to promote the division's surveillance and research findings on the impact of exposures during pregnancy. Her team also facilitates the translation of data into public health action on topics ranging from emerging threats during pregnancy to prenatal substance exposure. During the CDC's COVID-19 response, Ms. Pollan led communication for the maternal immunization team on CDC's vaccine task force. And with all of that, I'm really excited to get into today's event, and I'll turn it over to Dr. Chambers, who's serving as our moderator today.
Thank you, Katie. I really appreciate it. And I really want to extend my appreciation and thanks to the Society for Women's Health Research and the Coalition to Advance Maternal Therapeutics for organizing this webinar and the invitation to moderate the second in what I think is a series of 3 briefings on this really important topic of inclusion of pregnant and lactating persons in research, and in particular clinical trials involving therapeutics. So I'll preface the panel discussion with a few brief remarks by saying I have spent the last 30 years of my career working within our national network of mother-to-baby information services that provide individualized counseling and risk assessment for pregnant and lactating persons. And in this setting, we respond to tens of thousands of pregnant and lactating persons who contact us each year wanting to know if the medication or the vaccine they've taken or plan to take is safe for the baby. Unfortunately, it is still the case that for many of these therapeutics, typically newly marketed ones, but even for older products, that there is no or inadequate information that can help that family and that their healthcare providers make truly informed decisions about the best direction and best choices for that particular situation. At the same time, I've spent the last 30 years in what I guess I would describe a kind of an uphill battle of trying to obtain support to really develop that needed evidence-based information through what has been the kind of traditional approach, which is observational studies of pregnant and lactating persons and their children. So in other words, a mom who has already taken a medication or received a vaccine and is willing to be studied in terms of the outcomes for that pregnancy and that baby, but not participating in a randomized clinical trial where a mom might be assigned to receive a medication or not. I've done this through our national efforts, including mother-to-baby pregnancy studies, many of which are included in the Vaccines and Medication in Pregnancy Surveillance System, which is an initiative that it intends to try to move this field forward as quickly as we can. And then, as mentioned, through our human milk research biorepository study, where we do lactation research across the US. We continue to make progress, but I will say clearly it's not enough. From the speakers on today's panel, I know you're going to hear more about how critical this need is and the challenges that we face in moving this field forward appropriately and as quickly as possible. I'll end by putting this in a little more person-centered perspective. Every day, at Mother to Baby, the counselors across the network talk to people who contact them who say in so many words, I just found out I'm pregnant and I'm so worried that my asthma medication, the antidepressant medication I take, the treatment I need for my psoriasis, on and on, fill in the blank, could have harmed my baby. So what a dilemma to be pregnant, have already had an exposure to a medication and be concerned that something may have happened or there may be a risk associated, and to be in a position of not being able to have the adequate or appropriate information to provide that woman, that person, what she needs to know at that point in time. We also receive commonly calls from individuals who say, I'm planning pregnancy and I have Crohn's disease and the only drug that works for me is drug XYZ, but I'm afraid to take it during pregnancy because I've been told there are no studies about its safety in pregnancy. So what a dilemma again. Here's a situation where someone is trying to be forethinking about a future pregnancy, but is in a predicament in terms of what to do about being able to continue a needed and effective medication. And on the lactation side, every day we get calls from moms who say, I won't take this antibiotic or I won't get a recommended vaccine because I'm breastfeeding and I'm worried that that we don't know enough about this and that it may cause potential harm for my breastfed baby. Or, and maybe even worse, they sometimes say, because I need this medication, I will have to stop breastfeeding. And of course, neither one of those are the solutions that we hope that breastfeeding, lactating moms have to be faced with. So I think the good news is that this is a situation that is fixable. And if we can expand inclusion of pregnant and breastfeeding persons in research, including under appropriate circumstances in clinical trials, early in the life course of drug and vaccine development, we can move this field forward in a much more aggressive fashion. So I'm very honored to begin this session with our first speaker, Dr. Ann Lyerly. I will turn it over to Dr. Lyerly.
There we go. I'm sorry, I keep trying to put my video on and it keeps turning off. Sorry about that. I'm going to share my screen here. So I wanted to first of all thank Katie Schubert and Dr. Chambers, and to echo Dr. Chambers, I am both delighted and honored to be here. I want to thank the organizers for including me and recognize the really tremendous leadership of the Society for Women's Health Research over the decades, as well as the Coalition to Advance Maternal Therapeutics in supporting the health of pregnant populations and children. So I'm going to talk today about some of the consequences of our failure over the last several decades to include pregnant persons in biomedical research. In the words of the organizers, how leaving these populations out leaves them behind. My comments draw on about 15 plus years of work in this area, which includes the Second Wave Initiative, an effort I co-founded to ensure the health interests of pregnant people are adequately addressed in biomedical research and drug and device policies, as well as the Phases, Prepare, and Prevent projects addressing ethical inclusion of pregnant persons, specifically in infectious disease contexts. So what brought me to this topic, and indeed the foundation for any argument to address the challenges of including pregnant people in research, which were explicated so nicely in the previous session, is that patterns of exclusion, which is our status quo, have resulted in profoundly harmful evidence gaps, which I think about in three areas. The first relates to dosing. Clinically actionable pregnancy-specific data to guide dosing in the specific context of pregnancy are all but absent for most drugs. The second is fetal safety. For at least 9 out of 10 drugs on the market, we lack adequate information about their potential to cause fetal harm. And the third is maternal outcomes. Research in pregnancy often focuses on pediatric or neonatal outcomes, which means that we lack data on the impact of interventions on maternal health. These gaps have significant costs. Indeed, while excluding pregnant women from research prevents risk in research contexts, the risk doesn't disappear. Instead, risk shifts to the clinical settings where they expand. I call this risk shifting. So sort of in the next piece of my talk, my comments, I'm going to highlight 3 kinds of harms that result from this shifting of risk. There are more, but these are 3 that are particularly compelling, I think, from a public health and ethics standpoint. But I always have to sort of recognize at the forefront that that really suffering of pregnant persons in the face of uncertainty is a leading concern. So the first harm I want to talk about is ineffective treatment. So pregnancy is no magic bullet against medical illness. Pregnant women get sick, and women with chronic diseases get pregnant. They need drugs that will effectively treat their disease, and yet due to evidence gaps to inform the dosing of drugs, we can't ensure we are doing that. Pregnant bodies are different, physiologically different, with changes to the cardiovascular, gastrointestinal, renal, and enzymatic systems that change, sometimes dramatically, the way that the body processes drugs, or the pharmacokinetics. Without pharmacokinetic data, pregnant persons may be given the wrong dose so that they are either undertreated and remain exposed to the harms of disease, or overtreated, exposed to excess toxicities of drugs, and in many cases, they can't tolerate those drugs and remain exposed to the harms of untreated disease. Such errors have real consequences. Take for a recent example, the drug cobicistat, an HIV booster drug, which is paired with antiviral drugs that fight HIV in order to make those drugs more potent. Originally approved in 2012, it was widely prescribed, including to pregnant persons. Then in 2018, a pharmacokinetic study showed alarmingly that in pregnancy, concentrations of cobicistat and the drug it was boosting were dramatically reduced, up to 50%, raising concerns about viral breakthrough, resistance, and vertical transmission, or transmission from the mother to the baby. Note in these two graphs, the red arrows pointing to a large difference in drug concentrations between pregnant and non-pregnant persons. An important finding and research effort, I want to call attention to the delay. For 6 years, pregnant women were being prescribed a drug combination that was potentially ineffective in treating their disease or in protecting their offspring. The FDA now recommends against use of this drug combination in pregnancy. Harm number 2. So the second way that pregnant women are left behind is that they cannot and do not presume that the drugs they are prescribed are safe. In other words, the failure to do research leaves pregnant women who need to use medications potentially exposed to unacceptable risk. First, again, because most drugs come to market with only animal data, and the vast majority of drugs have undetermined teratogenic risk. The drugs that women are prescribed might have harmful effects on the fetus. Moreover, given gaps regarding maternal outcomes, the handful of medications deemed safe, or at least which obstetricians are comfortable prescribing for the fetus, may carry inappropriate risk for the pregnant person. So, of course, the paradigmatic example of unacceptable risk to the fetus is what happened with the drug thalidomide. And this was discussed in the last panel, but I need to return to it because the shadow of thalidomide has cast is so imposing. So to review, if you were here before, thalidomide was a drug developed for the treatment of morning sickness in the 1950s. It was approved for use in more than 20 countries, among them Canada, Europe, and Australia. And by 1962, the drug was linked to severe birth defects, limb reduction abnormalities called phocomelia. It affected more than 10,000 pregnancies worldwide, but approval was blocked in the US by FDA pharmacologist and physician Frances Kelsey, who was famous for standing firm in her conviction that data were insufficient to justify approval. For her efforts, she was given the President's Award for Distinguished Civilian Service by John F. Kennedy. So I tell this story because the thalidomide disaster is often cited as a reason not to do research. We don't want to have another thalidomide, is what people say, which is, to put it mildly, exactly the wrong lesson. Thalidomide was a result of inadequate research in pregnancy. In fact, it is a point not lost on Frances Kelsey herself. In her papers, which my colleague Miranda Wagoner pored over at the Library of Congress last year, and we published a paper on them, Kelsey noted, that the effects of thalidomide should have been recognized in a well-controlled clinical study involving comparatively few patients in pregnancy, thus averting the wide scale of the tragedy. So this is true because thalidomide was such a strong teratogen. For less strong teratogens, relatively few patients may not be a realistic cohort, but just to say for another thalidomide, that would have been effective. Kelsey also emphasized the importance of research. Conduct of clinical trials in pregnant women present special problems, but are necessary to assure safe and effective drugs will be available for use. So in a recent paper, Dr. Wagner and I offered that following Kelsey, the logical policy response would have been to pursue research with pregnant people, not forbid it. So there is still time, but we have lost some, needless to say. Okay, harm number 3. The third way that pregnant people are left behind is that fetal safety concerns make policymakers and doctors reticent to recommend and women reticent to use medications and vaccines, as Dr. Chalmers so eloquently described. Such reticence may harm both pregnant person and fetus. Harms of undertreatment may outweigh risks of medication use. And it's important to note that disease itself can be a potent teratogen. So there are many, many examples of the harms of reticence, not least of which is the vaccine reticence that followed failures to include pregnant persons in the COVID research agenda, which Dr. Poland will be delving into later. So I'd like to go to a different kind of classic story of reticence from another era, which is rubella. Though rubella is in many ways behind us, I consider it a cautionary tale, one which made clear the potential costs of caution. Rubella, or German measles, is an acute contagious virus that causes mild illness in adults. Infection in pregnancy causes congenital rubella syndrome, which can result in stillbirth and severe birth defects. To address an epidemic of rubella back in the 1960s, a live attenuated vaccine was developed, but due to theoretical risk of harm from live vaccines, the US strategy was to vaccinate around women of childbearing potential as well as pregnant women and focus on vaccinating preschool and elementary school age children, so to steer clear of pregnancy with as much of a barrier as possible. And yet there were 2 harmful, unintended consequences of caution. So forgive me for this complicated graph, but if you look at it long enough, it makes a lot of sense. So first, the caution was the use of caution here was ineffective as a public health strategy. So vaccinating children had the paradoxical effect of increasing the average age of infection. And so you can see the blue line, the average age of infection in the childbearing population. Surveillance data showed after the strategy had been rolled out for a bit that while rubella cases declined overall, cases among women of childbearing potential increased. Rates of congenital rubella were unchanged. So they changed their strategy, started vaccinating women of childbearing age, not pregnant women, but women of childbearing age, and congenital rubella declined and has remained virtually undetected for decades. So you can see where that blue line dips down in between 1977 and 78, and that was the effect of, loosening the grip on this caution that had been deployed. Second, there were other consequences. Although rubella vaccine has never been shown to cause congenital rubella, it has remained contraindicated in pregnancy, and many exposed pregnancies have been ended over fear of what the adverse effects of rubella vaccine exposure might be. Caution has consequences, and they are not always the intended ones. So in summary, without adequate evidence, pregnant persons may be given the wrong dose so that they are either undertreated and exposed to disease or overtreated and exposed to toxicity. They may be given drugs with unacceptable risk, or they may be denied access to beneficial drugs. There's one more harm that relates not to evidence gaps, but to the immediate benefits of participation in that research participation may provide. So some clinical trials offer the prospect of direct benefit, but pregnancy can be a barrier to accessing such benefits. Particularly harmful, these are particularly harmful where treatment options are limited or the condition is life-threatening. Exclusion leaves pregnant women and offspring exposed to life-threatening illness. So one heart-wrenching example of this harm was the Ebola epidemic of 2013 to 2016 in West Africa. Ebola is a life-threatening infection with high mortality rate, especially in pregnancy. At the time, 89 to 93% of pregnant women who were infected died, and practically all of their infants. Despite the urgent need both for trial access and for data on care of the pregnant population more broadly. All drug and vaccine trials excluded pregnant people. Some offered that this was an example of pregnant women being protected to death. So much of what we have discovered in our efforts is that exclusion of pregnant persons is a product of habit, entrenched patterns of reasoning, and misunderstandings of what it is that ethics demands of the research community and those who regulate it. Part of why I believe that progress is being made is that the cultural shift that PregLac called for in its report seems to be taking hold. Indeed, 3 conceptual shifts have emerged in the ethical framing of research with pregnant persons, which I think are quite powerful. The first is a shift away from the historical designation of pregnant persons as a vulnerable population. This is a designation shared by children, prisoners, and individuals with intellectual disabilities. Not only is it inaccurate, nothing about pregnancy itself interferes with a person's ability to provide valid consent, and I've made this case while pregnant before, but it has had an unintentional chilling effect on research. In 2018, the Common Rule dropped pregnant persons from the list of individuals considered vulnerable, and the American College of OBGYN and others designate pregnant populations as complex or special. The second is a shift away from an emphasis on protecting pregnant persons from research, of eliminating that tiny risk bubble in the research context and protecting them through research. Exclusion from research has put pregnant populations in harm's way, and so the best way to advance the health of pregnant populations is to generate data to inform their care. Third is the shift from presumptive exclusion to fair inclusion. So pregnant persons deserve both a fair share of the benefits of the public and private investments in research and fair access to trials, especially those like Ebola. 3 ethical foundations are important to identify that undergird the responsibility to address inequities in the evidence base, and I'll quickly go through these. First, equitable protections from research risk. It is the responsibility of the FDA, they state it, to protect the public from harmful drugs, and an animating mission of research to gather evidence in controlled and regulated contexts to decrease risks in clinical care settings. Pregnant persons deserve this protection alongside other populations. The second is access. Pregnant persons deserve timely access to first-line and optimal medications, and the absence of data has been a barrier to that access. The third is respect. When research is conducted, attention to fetal and child outcomes can overshadow attention to maternal health outcomes. Research involving pregnant persons must engage them as ends in themselves and prioritize their health interests as key study outcomes. So I think it's clear that leaving pregnant populations out of research leaves them behind. And so I'm going to end with a quote from Frances Kelsey. "It would indeed be unfortunate if fear of adverse effects to the offspring deprived the mother of drugs that might be essential to her well-being and indeed possibly also to the successful outcome of the pregnancy itself." Thank you very much.
Thank you so much, Dr. Lyerly, for that terrific overview. And now we're going to hold on questions and have Kara Pollan speak next.
Great. Good morning, everyone, and thank you for the invitation to speak today. My name is Kara Pollan, and I am the Associate Director of Communication for CDC's Division of Birth Defects and Infant Disorders. And I'd like to tell you about our experience of making public health recommendations in the context of limited available data for pregnant people through the lens of the maternal COVID-19 vaccination experience. Next slide, please. When COVID initially hit in early 2020, we really weren't sure if infection during pregnancy would lead to severe illness and pregnancy complications. However, over the course of 2020, sufficient data had accumulated demonstrating that COVID-19 did indeed present increased risks during pregnancy. By fall of 2020, we knew that COVID-19 infection during pregnancy was associated with increased risks for ICU admission, invasive ventilation, including ECMO, and death, as well as pregnancy complications and adverse pregnancy outcomes. Next slide, please. In mid-October 2020, early conversations began around how COVID-19 vaccination recommendations might apply to pregnant populations, specifically healthcare providers who would be in that first phase priority population for the vaccine rollout. We expected about 330,000 healthcare providers to be pregnant or recently postpartum. For the initial COVID-19 vaccine clinical trials, pregnant people were excluded because of theoretical risks to the fetus. Therefore, in the absence of data, we discussed possible scenarios. So we discussed whether to include pregnant people, we discussed whether pregnant people would be excluded from being offered vaccine while safety data accumulated, or whether to be permissive with shared decision-making and really encourage decision-making with discussing the risks and benefits. Next slide, please. When COVID-19 vaccines were authorized for use in December 2020, we had very limited safety data available about use in pregnancy. We had some data from DART studies conducted by the pharmaceutical companies, and no safety concerns were noted. We also consulted with OBGYN experts who agreed that mRNA COVID-19 vaccines were unlikely to pose risks to pregnant people or infants. Despite very limited safety data, pregnant people were included in ACIP's recommendations with this language. Pregnant people who are eligible for COVID-19 vaccination can choose to receive vaccine. Importantly, CDC ensured pregnant people had access to COVID-19 vaccines and included specific language that vaccination did not require approval from a physician. Next slide, please. In early 2021, as the vaccine rolled out, states varied in their approach to prioritize pregnancy as a qualifying condition for vaccination. In February of 2021, 15 states included pregnancy in phase 1, while 35 states and DC did not include pregnant people among their priority populations. Next slide. At this time, misinformation rapidly spread. Rumors circulated widely on a range of topics, but for this population specifically, myths were rampant that COVID-19 vaccines led to infertility, miscarriage, damage to the placenta, and more. CDC proactively disseminated messaging on social media and other digital channels, engaged with providers and other trusted sources to share accurate information, and developed resources and worked with partners to combat this misinformation. Furthermore, inconsistent guidance globally caused confusion among pregnant people and their families and might have increased mistrust or skepticism in this population. Next slide, please. At the same time, in the absence of clinical trial data, CDC leveraged existing safety monitoring systems and rapidly launched new systems to monitor and follow up on the health of pregnant people who received COVID-19 vaccines and their babies. On this slide are some of the tools that CDC uses to keep close tabs on the safety of COVID-19 vaccines. So vSafe was launched to collect safety data in real time after someone received a COVID-19 vaccine. If you enrolled in vSafe and reported that you were pregnant at the time of vaccination or after vaccination, CDC could contact you for enrollment in the vSafe pregnancy registry. And this is where we collected more information about pregnancy and infant outcomes following COVID-19 vaccine. Other systems shown were existing systems that CDC leveraged to monitor safety data. Next slide, please. In April, safety data began to emerge when CDC released the first US data on the safety of mRNA COVID-19 vaccines administered during pregnancy. And these data showed similar side effects reported by both pregnant and non-pregnant women after vaccination and did not identify any safety concerns for pregnant people who were vaccinated in their third trimester or for their infants. As spring turned to summer, reassuring data continued to accumulate, not just from CDC, but more broadly from data published around the world. Multiple studies assessed vaccination early in pregnancy and did not find an increased risk for miscarriage among people who are vaccinated before 20 weeks of pregnancy. Studies also found that vaccination was associated with a significantly lower risk of severe illness for pregnant people. And lastly, studies demonstrated detection of maternal antibodies and umbilical cord blood, indicating the potential for protection against COVID-19 for neonates and infants. Next slide. In August of 2021, given the increased risk for severe illness and death from COVID-19 during pregnancy and the accumulation of data on the safety and effectiveness of COVID-19 vaccination during pregnancy, CDC made a strong recommendation for COVID-19 vaccination in pregnancy. This was about 8 months after COVID-19s were initially authorized. This strong recommendation aligned with recommendations released from several clinical organizations. And we really hope that this unified recommendation conveyed a strong, consistent message about the life-saving benefits of vaccination before and during pregnancy. Next slide. Around this time, though, cases started increasing again. A clear fourth wave of infection emerged with the Delta variant. We began seeing more than 1,000 cases reported among pregnant people each week. Next slide. As the Delta variant surge continued, we observed severe outcomes increase among people who are pregnant. So we observed an increase in ICU admissions and deaths, specifically in August, which had the highest number of deaths in this population during the entire pandemic. Additionally, pregnant people with COVID-19 during the Delta period had 4 times the risk of having a stillbirth compared with other time periods. Next slide. These numbers represent real people. This is Kia Slade. She was 7 months pregnant, unvaccinated, and fighting for breath when her son came into the world. She had to have an emergency C-section at her bedside in the ICU, delivering her son Tristan 10 weeks early at just 2 pounds, 14 ounces. Kia didn't meet her son for 2 months since she was on a ventilator and a medically induced coma for 8 weeks. It was only after a perilous 2.5 months in the hospital that Kia was vaccinated against COVID. In this article published by Kaiser Health News, Kia said, "I wish I had gotten the vaccine earlier. Doctors kept pushing me to get vaccinated, but there just wasn't enough information out there for me to do it." Next slide, please. In September, CDC released a health alert notice strongly recommending COVID-19 vaccination either before or during pregnancy. This notice highlighted the increased risk from infection and the reassuring safety data that had accumulated about vaccination. We also provided tangible actions for public health and healthcare providers to help improve uptake. Next slide. As we moved into fall, however, coverage of COVID-19 vaccination among pregnant people remained low compared with non-pregnant people. This graph depicts coverage of COVID-19 vaccines among pregnant people reported to CDC's Vaccine Safety Data Link. As of November of 2021, so that was fall of that year, about 35% of pregnant people had been fully vaccinated against COVID-19 prior to or during pregnancy. Next slide. Vaccine coverage also differed by race and ethnicity. So as shown in this graph, the highest uptake was observed among pregnant people who are Asian non-Hispanic at around 50% that fall. And the lowest uptake was observed among Black non-Hispanic pregnant people around 20%. Next slide. Efforts to improve maternal COVID-19 vaccine uptake are ongoing. We continue to maintain several resources that providers and partners can use to increase vaccine confidence and uptake. We at CDC continue to work closely with our partners to engage clinical organizations, streamline access to vaccination, and amplify messaging to pregnant people and their families. Next slide. The last point I want to highlight is that vaccine hesitancy around COVID-19 vaccines have likely contributed to lower uptake of other routinely recommended vaccines. For example, as of last month, nearly 50% of flu-related hospitalizations in women of childbearing age have been in women who are pregnant. In addition, flu vaccination coverage among pregnant people was more than 10 percentage points lower than the previous year, and more than 20 percentage points lower than it was prior to the pandemic. Next slide. Great. So to conclude, I'd like to highlight some of the key themes and lessons learned from our COVID-19 experience. Participation in VSafe demonstrated that pregnant people at risk for severe illness want access to new vaccines and therapeutics. Vaccines were offered and accessible to this population, but without safety data, we lacked a strong recommendation for COVID-19 vaccination for 8 months. This led to states differentially including pregnancy as an eligible condition, as well as providers giving inconsistent guidance to their patients. Misinformation is a powerful deterrent to implementation of vaccination recommendations for pregnant people, and without safety data, it really took hold. Low uptake of vaccination persisted over time, and deeply rooted safety concerns likely contributed to this, especially among groups at greatest risk for severe illness from COVID-19 infection. Vaccine hesitancy about COVID-19 vaccines in pregnancy likely contributed to lower acceptance of other vaccines as well. Thank you.
Thank you so much, Kara. That's a perfect story for this topic. So I think we're going to bring the two panelists back on, and we'll open it up for questions. I will go to the Q&A. If you can put your questions in the Q&A, we'll start working through those, and we have a couple that are already on board. The first one comes from Tacey White, stating that Dr. Lyerly stated that 90% or more of drugs have uncharacterized teratogenic risk. Tacey works in this area, so she says most of these drugs have been specifically studied for teratogenicity in 2 separate animal species, as is required in preclinical testing, and typically at higher doses than are used clinically. So clearly there is a need to study these drugs in pregnant people, but how can we increase the use of animal data as part of the human risk assessment? Do you want to make a comment about that, Dr. Lyerly?
Sure. And I mean, I think Dr. Chambers, I would love to hear your thoughts on this as well. But I think it's just important to note that while animal data are required and they're important and they're in place for most drugs, they are not the greatest at predicting what kinds of impacts drugs will have on offspring. So you can have evidence that you can have no evidence that there's any impact, and you can find out that there are abnormalities, and you know, the reverse is also true. You can have you know, risk signals in the context of animal studies. They can worry doctors and patients for a long time, and then when you finally get the population studies done, they don't pan out to be true. So you know, there is just an urgent need to get good data in human populations. But the animal data are important, they're necessary, but not sufficient.
So I'll just add to what you said, Dr. Lyerly, and I would agree completely. I do think, as Dr. White is saying, that sometimes the value and the information that the preclinical studies or after a drug is marketed from animal species can provide, sometimes it gets lost in translation. And so on the, you know, if you want to call this a positive side, we certainly see new medications. We've had some recently where the animal data clearly show a signal, and this will just not be used in pregnant women at all based on that strong signal in the animal data. So it can kind of drive, you know, putting a curtain in place early on in the process, but it's the much more sort of vague area where there may not be a signal, there may not be a strong signal, and then we're trying to use that data to try to inform human use. And ultimately, human use is needed to flesh out the answers to these questions. So I will take the next question. Coming from Lauren Kozlowski, and I think this is probably for Kara. I hadn't seen the numbers for how much flu vaccination has gone down following COVID. Not surprising, but very concerning. Just a comment. Thank you for this presentation. So Kara, any thoughts on, I think we've seen a downtick in child vaccination in the pandemic. Go figure.
Yeah.
Yeah,
I think vaccine hesitancy has contributed to low uptake for not just pregnant populations, but we're also seeing it in the pediatric population. So there's a lot of effort across CDC to really highlight these concerns and do what we can to ramp up vaccination and make sure people are aware that vaccination is beneficial and can really protect not just children, but also pregnant people during pregnancy. So.
And a follow-up on, I think the kind of real life case history of what happened with COVID-19 vaccine is so illustrative of the dilemma. If we could turn back time and when these new vaccines, particularly the mRNA vaccines, were developed, had there been even a very small clinical trial in second and third trimester, not first, for pregnant women before the emergency use authorization, even that tiny bit of data, do you think that would have made a difference in the messaging, both from the authoritative bodies, but from clinical comfort and being able to recommend the vaccine? And I'll ask that to both of you.
Yeah, I mean, I can jump in with this. I was pregnant during the pandemic, and so this situation and story like feels very personal to me. So I was pregnant before vaccines even rolled out, but I think even if we could have had messaging to say that there is some evidence, we don't have a lot of it, but some evidence that is reassuring, I think it could have changed the way we framed it. But having to say, you know, we don't have much at all, you have to sort of make your own decision, I think was a struggle for a lot of people. And then I think that also trickled down to how providers were counseling patients. We've heard stories where people were recommended to wait until after the first trimester or wait until after pregnancy. So lots of variability in how guidance was applied or provided to people who were pregnant at the time.
Right.
I mean, I would just add that it is a confusing message that it is, you know, inappropriate to enroll a pregnant person in a study where you're watching them very closely and collecting data, and yet it is okay for that person to get the drug or vaccine outside of the context of research when you still don't know whether there's risk, right? So I think for some people that was sort of that there was a confusing sort of calculus there, and you know, I think part of going back to the animal studies, from my understanding, part of the reason that pregnant persons were not enrolled in those studies was that the animal studies weren't done in time, which is an argument really for getting those done earlier. And I think there's a critical need across diseases to move up the conduct of those DART studies.
Yeah, and then in this kind of compressed time situation with the vaccine, with the rush to develop them, I'll just add one more thing, and this is something that we dealt with counseling on vaccine, is in the, you know, not unexpected high level of concern about this, even with had there been, you know, a small number of people who had participated in clinical trials prior to emergency use authorization, this kinds of questions we got are the ones that you talked about, the misinformation. Well, how do you know it doesn't produce infertility? Or how do you know that the vaccine is not going to create, you know, a problem with the child with learning disabilities later on? And those are unanswerable questions for quite a long time, even if you do have trial data before a vaccine or a drug gets marketed. So the communication piece of this was big. And I'll just add one more thing and then go again to the questions. Are there, I was thinking, Dr. Lyerly, about your discussion about shifting of risk. And this was, I think, a perfect example where the risk got shifted to the clinician to make the decision. And, you know, what in your experience or judgment, what are the, you know, what's the risk calculation that the clinician has to consider in the absence of clear, you know, official guidance or clear data to support a decision?
Well, I mean, I do think it's a very difficult position for clinicians to be put in, because they don't have the data to guide care. So they need to bring their clinical experience into the room. They need to frame the decision for the pregnant person, which, you know, different people value different things. So that conversation can be interpreted very complicatedly, if you will. So I think it's a really sort of, I'll just say, unfair burden to put on doctors. And you know, when there is sort of admonition to consult with your doctor about whether you should do this or not when there is no data. It is, I mean, to use not a very high level term, it's in some ways a cop out. I mean, I think all of us can look at the risks of COVID. All of us can look at the risks of the vaccine, which were not known, but we did know that there was not even any theoretical risk for the mRNA vaccine, right? So to say, take it if the benefits outweigh the risks, well, you don't know what the risks are of taking the vaccine, but you know that there are benefits, especially with a functioning vaccine. So the fact that the physician has to interpret and convey that to individual patients is, I think, really, it is really problematic.
Yeah, and the liability issues as well.
Of course,
they
of
have
course.
those concerns. I'll go back to the Q&A. This is going back to thalidomide. Were there animal studies with thalidomide? Did they show a limb reduction issues? So there are others who are on this call who can probably answer this more clearly. But my understanding is that at the time when thalidomide was not approved in the U.S. for other reasons, but was approved in other countries, it was not known from animal studies, and apparently there are only certain species who are sensitive to thalidomide to thalidomide, but it also, because of the thalidomide issue, that's what led to the requirement for preclinical testing relative to new medications. I don't know if anybody has additional information, but that's my understanding. Next question is, I am a researcher focused on human milk and lactation from Warsaw Medical University in Poland. Because breastfeeding women are excluded from clinical trials, there are well-defined side effects of medical intervention. Are there well-defined side effects of medical intervention for lactation, or rather not? I'm not quite sure I understand the question, and I'm going to actually, if the questioner could add a little bit more clarity about what is meant by medical intervention, I'm going to leave that in the pool in the Q&A and see if we can get a little more detail on what it is you're asking. And I'll go on to the next question in the meantime. Do you think the better solution is including pregnant patients in pivotal studies, or to have separate pregnancy-specific studies that may address the dosing question? Good question. Anybody want to take that?
I mean, I'm happy to offer some thoughts about that. I mean, in general, we need all kinds of research. So there will be some contexts, generally speaking, where pregnant women should be included in a general population study. Maybe analyses of those pregnant people should be done separately. And there are some circumstances in which pregnancy-specific data need to be collected. For pharmacokinetics, I mean, there are people that do pregnancy pharmacokinetics. They are devoted, they do beautiful work, and it is in high demand, but we need more of it, and it needs better funding. But there's really amazing opportunities to gather those pregnancy-specific pharmacokinetic data opportunistically. When pregnant people take drugs, you can do a study where you measure drug levels without imposing any risk in clinical research. So those are considered minimal risk studies, and we consider them low-hanging fruit that just needs to be gathered. So, I think particularly for pharmacokinetics, doing specific studies directed at pregnancy is important, both sort of post-approval in these opportunistic settings, and then there's some really wonderfully ingenious research designs that can be done pre-approval if the motivation is in place and the DART studies are done. So but yes, for pharmacokinetics, really pregnancy-specific studies are what is needed.
So thank you for that. And I'll just add, I agree. I think the answer to that question is both, that we need to have multiple ways that we're gathering this information. both in the context of people in a clinical trial who become pregnant unintentionally. So carefully gathering that information in the context of where it would be appropriate. And there is now draft FDA guidance on inclusion of pregnant women in clinical trials and when it is appropriate, when they might be included as part of a general population. But definitely the pregnancy specific work relative to appropriate dosing. And as you say, I think PregLac, that's one of their recommendations, is to make sure that this type of work is supported, because it is incredibly important. I'm thinking of the, I mean, certainly this is being done for a lot of HIV drugs, but to make sure that the mom is getting the appropriate dose in pregnancy, which may not be the same when that person is not pregnant. I'm going to, in the interest of time, I'm going to ask this last question. Thinking about what was presented here today about inclusion of women or pregnant persons and lactating persons in research, and this is to both of you, what do you think are the reasons why a person may or may not be willing, a pregnant or lactating person? What would be the barriers to participation? And it could be lack of awareness, concern about posing additional risk, or maybe just nervousness about taking part in research. Those would just be some examples. So maybe I'll ask Kara. I know you had really good participation, I think, from VSafe in the pregnancy registry, but any thoughts about what may or may not motivate a pregnant or lactating person to participate in a research study?
Yeah, I mean, I think we had really great participation in VSafe because the initial rollout was healthcare providers, and they were frontline workers who are at risk of severe illness. I think they also may be more health literate and aware of what the risks are or the lack of risk, the risk of infection, and probably the lack of risk for the vaccine and the benefits of the vaccine. So I think they may have a better perspective coming into the registry. So I think that's why we had such great participation, especially at the beginning of rollout. In general, I think health literacy is an issue that we've seen magnified by the pandemic that we really need to address, and so that's definitely something we are trying hard at CDC to make sure our messaging is clear and easy to understand, and trying to see what we can do to make sure that information is accessible to people who are thinking about these questions, and trying to support the work that's being done.
Yeah, thank you. And before I go to Dr. Lyerly, I think you're actually responding to a question in the Q&A from Erica Hensley about research intersecting with a lack of health science literacy among the general population, and how we can do a better job in public health about moving the messaging forward. So to you, Dr. Lyerly, and thoughts about why people would or would not participate?
Yeah. So, I mean, I think there is a mythology in some ways that pregnant people or even women are unlikely to want to participate in research. I mean, that was one of the arguments against including women way back in the 1990s when they were underrepresented in research, as people on the Society for Women's Health Research will remember. When the Revitalization Act was passed and there was requirements for inclusion of women, now we have more than 50% of research participants being women. So the problem wasn't willingness. The problem was on the part of the sort of research infrastructure and, misperceptions about what ethics requires. I've done many studies interviewing pregnant people or people who have been pregnant about whether or not they would participate in studies, or in retrospect, why they did. And, certainly there are some people, pregnant or not, who are not going to be interested in participating in research for a variety of reasons. But I will also say that pregnant people, first of all, are aware of the idea that clinical care is like uncontrolled phase 4 research, right? You are taking a drug that has not been tested, and yet when you do that in clinical medicine, you're not being watched and data aren't being collected. So some people understand that taking medication or vaccine in the context of a study, which is something they'd be taking anyway, is, in their best interests, right? So I had one interviewee say, I think it was, I felt safer and more protected in the context of research than I did in my clinic under the care of my doctor. I mean, that's great. Not all women will feel that way. But the other piece is that, pregnant people as a population recognize that these evidence gaps exist, and pregnant people, like others, want to help. They want to help grow the knowledge base so that other people don't suffer with the uncertainty the way they have. And I think we have to give credit to the idea that there is that form of, generosity and meaningfulness in contributing to the research.
I firmly believe there's nobody more altruistic than a pregnant or breastfeeding mom. So in our last 30 seconds to both of our panelists, can you say what is the one thing you would like the attendees of today's webinar to take home from this discussion? Kara?
Yeah, the one thing, I should have been prepared for this question, Tina, but I think just that the experience of COVID-19 vaccination really highlighted a lot of key themes for us. And I think having that data initially around safety and effectiveness could really have helped frame our messaging as we rolled vaccine out. And hopefully we can get the word out that there actually is a lot of data now, and it is safe and effective for use during pregnancy.
Not too late.
Yeah, not too late. Not too late.
And Dr. Lyerly, last one take home.
Sure. Well, I mean, I come to this space from the standpoint of ethics, most primarily. Ethics has often been used as the reason that we don't do research, but I think it's very clear that there's an ethical imperative to do research. So now it is the responsibility of, leadership to make sure that these studies are funded and able to move forward so that we take care of pregnant women and their offspring.
And I'll close this by echoing what you both said, that the most important take-home message is that this should and must become a priority for us as a human population. So I want to thank everyone. I don't know, Katie Schubert, if you want to give any closing remarks, but I want to thank everyone for attending and thank our panelists. You did a fantastic job of a difficult topic area.
Just a big thank you to all of you. This has been really informative, and I would encourage this tends to be a pretty hot topic, I think, which is really encouraging. Continue to join us. We have one more webinar in this series, but the work will continue through the coalition to make sure that we have better inclusion of these populations. So thank you all again.